Good evening. Welcome to the German Center for Research and Innovation. Welcome to the German Consulate, the German House. It's really good to see so many of you here tonight. With the torrential rain pour earlier, we were worried that it was just us talking about aging tonight, but this is a really good turnout. Thanks all for being here, and also it's good to see so many familiar faces. Since this isn't our first aging event, but Eva will talk about that more later. My name is Gerrit Rössler. Many of you know me from previous events over the last year. I took over a year ago, almost to this day, the German Center for Research and Innovation as its manager. And the German Center for Research and Innovation is the host of this evening tonight, together with our partner organization, the University of Cologne. The German Centers for Research and Innovation, there's 5 of us worldwide. The others are in Moscow, New Delhi, Tokyo, and Sao Paulo. In fact, I had the pleasure of doing a 7 a.m. conference call with all of them this morning, because you can imagine with a worldwide network, you cover a lot of time zones. But it was worth it, because this worldwide network is supporting German research and innovation worldwide, trying to create visibility, trying to create exchange and dialogue. And tonight we'll see a wonderful example of that with various nations involved, even beyond Germany and the United States. So that's very exciting. We have, so an event like this, one doesn't organize alone. So Eva and I had the great help of our team here at the German Center. You've met some of them downstairs already. Some of them are in this room. If you see any of the team members, tell them thank you very much, shake their hand, because they're doing a fantastic job always putting these events together. And I just roll in and grab the microphone. So thanks to the GCRI team, Nadia, Luisa, and Jared, and Emily. Thank you very much. So, as the GCRI, we have a couple of exciting things coming up that I just wanted to let you know about real quickly. We have, in just a few weeks, we have an event, so actually in October, but still that's just a couple of weeks from now. We have an event on artificial intelligence and machine learning, cross-modal learning with the University of Hamburg. That's very exciting. And we have a larger event on virtual reality and augmented reality in Soho at the RISE Center. You'll all receive emails about that. Very, very exciting stuff coming up. And if you subscribe to our newsletter or our event invitation list, you'll learn all about that very soon. Oh, and a personal highlight of mine, at the end of October, we will relaunch our website, which is good news for everybody. It's going to be a barrier-free, bilingual website that contains all the information that we already have in a much more organized, much more intuitive and comprehensive way, so that you can find our events more easily and can go through our innovation with a lot more, with a much better experience. So look forward to that, and once it happens, make sure you let everybody know. So, without further ado, again, an event like this one can't always organize alone. I already mentioned my team, but I also want to mention the wonderful cooperation with the University of Cologne New York office. And this is maybe a great point to hand over to Eva, and I wish you all a wonderful evening and a wonderful discussion. Thank you very much.
Good evening, everyone. Thanks, Gerrit. Welcome to the German House. My name is Eva Bosbach. I'm the Executive Director of the University of Cologne New York office. You might have heard about Cologne, about the Cologne Cathedral. You might have also heard about the University of Cologne, which is one of the leading German research universities. In fact, it's one of only 11 German so-called universities of excellence. It's also one of the oldest higher education institutions Europe, founded 1388, and currently also the largest university in Germany. We have almost 50,000 students. The University of Cologne holds representative offices in China, India, and North America, here with our New York office. We are very happy that tonight we have Christiane Biel here, who is in charge of partnerships with North American universities from Cologne. She is here for the week, so if you would like to partner with us, please talk to Christiane here in the red scarf afterwards at the reception. Thank you so much for being with us tonight. I also want to thank to the German Center of Research and Innovation and Garrett and his team for such a great partnership, and also for sharing the values with us and the vision that we want to promote transatlantic exchange of ideas and dialogue and cooperation, which we're trying to do with events like this. We have a series going on, Aging in Society, which we had 2 events last year where we looked at the topic of aging. Also, maybe some of you were here for those events. That was more from the ethical and societal perspective. And tonight we will switch the view a little bit, and we'll look at the aging and aging-associated diseases more from the biological and medical and pharmaceutical point of view. So live long and prosper. We all want that, and I'm personally looking forward to the secret or maybe public recipe how to do this. And we're very happy that we have Molly McLeod tonight with us as the moderator of the evening. So I would like to introduce Molly to you first, and then the speakers will take their places, and then Molly will guide us through the evening. So I think personally that Molly is the perfect moderator for such an event, because she's a scientist and a science communicator. So she has a PhD from Rutgers University and then worked in creative and scientific writing, and now is working for the worldwide research and development communications team at Pfizer. And if I understood correctly, Molly, what you do, basically you go to see scientists and see what great things they do and what great discoveries and innovative ideas they have, and then you communicate that to the wider audience. So I think that's perfect for tonight, and I wish us all a great evening, and thank you all for being with us tonight.
Okay, it's fixed up. Thank you so much, Eva and Garrett. I'm really happy to be here. So, I think we all have a lot to learn from our panelists, and this is a very timely discussion because, it's not new for people to wonder about aging. But what is new, I'd say, in the last several years is really the depth of our biological understanding in what drives aging, what does it mean, how does it affect us, and then what do we do about it? So I have a lot of questions, as I'm sure you all do as well, about that science and what it means about, as Eva said, the recipe for longevity. So welcome to our panelists. So we have Jan, Bjorn, and James here tonight. So Jan's research focuses on the relationship between damage to the genome and aging. He's professor and chairman at the Department of Genetics at Albert Einstein College of Medicine. Bjorn's research also focuses on mechanisms of DNA damage and repair in aging-related diseases, and he's currently the director of the Institute of Genome Stability at the University of Cologne. And then James comes to us from Apollo Ventures, which is based in Hamburg, and this is a company that he launched to support biotech entrepreneurs who are looking for the next generation of therapies in aging-related diseases. And he also has a background in related research from UT Southwestern, where he got his PhD. So welcome. Thanks for being here. Yeah. So I think, as I said, it's a really timely discussion right now. So let's start by just level setting from, why we're here right now. Because it really seems like aging research has caught the attention of the public in recent years. So why has that happened? What is that new? What happened to bring that on?
I think there was really a revolution in our understanding of aging, and I think several things came together at this time, and this is why this is so timely. On the one side, we learned through aging research, which is still a very young and new discipline, in fact, that aging is ameliorable. And by understanding that there are genes that influence aging and there are damage that causes aging, we have for the first time the opportunity to actually target the aging process. And parallel to that, what happened is that the demographic change really results now in a life expectancy that is really unprecedented. So I think it's just so, like, almost every one of us is faced with a long life, which also comes invariably with disease. And so I think it's a huge societal topic. It's for more people than ever in the history of humanity, or a personal issue, and at the same time, the science is really taking off.
So just jumping in here, I would echo Bjorn's comment that there's been, I think for the first time, a critical mass of research from kind of different strands of science that have really weaved themselves together to a comprehensive understanding of aging. Some of the topics that scientifically we can discuss today, like genome damage and so on, we've known played a role in aging for quite some time, as well as like free radicals, which many of you may have heard about, and antioxidants that have some role in aging. But our depth of understanding of these things has improved so dramatically with new tools that really started, I would say, around the Genome Revolution of the early 2000s. And in the pharmaceutical industry, we think about very long time frames of innovation, right? It takes a scientist working 10, 15, 20 years before their idea can make it all the way through validation in mouse studies and then finally reach a patient in the clinic. And so sitting here in 2018, this is kind of like, in my view, some of the first fruits of the genome era that started back in the early 2000s when we sequenced the human genome. And so I think it's really just the beginning of a wave that will change the way we do medicine in a big way.
So sometimes we forget that half a century ago, when you were talking to your colleagues, to scientists, and you were saying that you were working on aging, they thought you were a charlatan. Nobody took you seriously. Of course, they forgot that a little bit earlier, not even that much earlier, maybe decades before that, when you were saying that I want to cure cancer, you were a charlatan. Cancer was simply there. It was unavoidable. That's what happened, and you shouldn't work on it. Of course, we started to work on cancer. I don't say we cure it now, but in fact, we did make a hell of a lot of progress. That's very clear now. You see that everything goes better. People survive longer. More people are being saved. You see the incidence going down. So I don't say that that will happen with aging also, but as they already pointed out to you, a lot changed, and we got more insight into what aging is. We sort of stopped saying that it's just inevitable, it just happens. Nothing just happens. You just think about the origins. Where does it come from? And those, I think, were the two main developments. First, people started to realize that they really had to find out why is there aging in the wild. Because if you take animals out of the wild, where they normally never become very old, of course, because they die from accidents or they're eaten up by other animals. But when you protect them, now you see aging in all these animals. So the question is, why does evolution, in all its smartness, why did that allow that? That problem has been solved, again, about half a century ago. The second thing is, can we do something about it? That problem seems like it has been solved also, since we found out, first people with nematodes, like Bjorn's favorite organism, they noticed that they could actually, when they dampened down, inhibited a little bit all sorts of growth functions and metabolic functions, which you would intuitively say that's not good, because the more growth, the stronger you are, the better. No, when you dampen that a little bit, suddenly they saw you could increase the lifespan dramatically. That's a major discovery. Now we know that not only the lifespan can be increased, but you see that the health improves. I think these two developments, that we understand why there is something like aging, and that actually it is possible to do something about it, those are the big differences. And now we realize that. That's why there's such an enormous enthusiasm now.
So would you say that one of those things is the biggest breakthrough, one of the biggest breakthroughs in our understanding of aging?
Well, the two biggest breakthroughs, yes, I just mentioned.
It can be one of yours too, one of your breakthroughs.
Well, it's really the evolutionary understanding that we now have plus the fact that we actually experimentally could demonstrate that we seem to be able to do something about it. I just say seem to because I think this should be a discussion point. I'm not so convinced.
Anything else? Do you think that you can name one of the biggest breakthroughs in our understanding of aging? Is it?
I totally agree with Jan. I would say that the most important breakthroughs are kind of 3 points. It is on the one side that we understood that the inevitable accumulation of damage drives the functional decline, the decline in tissue repair, in stem cells to regenerate organs, for neurons to degenerate and die in dementia. That is really the driving factor of aging. And from the evolutionary biologists, we understood that that is because our repair functions only work for 3 or 4 decades, because this is all that mattered in our history. So up until 150 years ago, human life expectancy was about mid-30s. If you eliminate from the calculation all the child mortality, you're at mid-40s. During that time, the next generation had to be installed. This is what evolution is about, getting the genes forward passing them on to the next generation. So all our genes are perfectly selected for keeping us repaired, maintained for 3-4 decades. Everything after that is extra. And so the second big revolution that Jan alluded to was about in the early 90s when the discovery was made that alterations in a single gene could double a lifespan of a nematode worm. Now you might think, okay, a nematode worm, what does it have to do with humans? We have the very same genes in us. And based on, and after that, over the past 25 years, we discovered more and more of these longevity pathways, genes that, when modulated, can alter life expectancy, can alter how long an organism is healthy. And that came then to the third point, that these pathways could be targeted. And the very first intervention that was actually already observed in the 1920s, 1930s, is that calorie restriction can extend lifespan. And what does that mean to us humans? Well, we all know that diets never work. So what we need there is actually interventions. And here a key discovery was that they're actually signaling mechanisms that respond to the calorie restriction, and they are responsible for the lifespan extension, for the gained health span. And so we can dissociate that from these from the dietary aspects and really develop now pharmacological inhibitors that can exert the same effects. And I think that's really the major breakthroughs where our whole concept about aging has changed.
I think this is a really fun way to start this discussion. And I wanted to kind of explore a little bit more Jan's point, talking about the foundation of our understanding of the evolution of aging, right? The question of why aging exists and what that means to the research today is such a fundamental and interesting question, I think, to explore a little more. So the way that I kind of conceptualize this is that evolution is a really harsh mistress. So as you said, evolution just doesn't care about us. After we get 4 decades on, then things in our bodies stop working because we supposedly, at least we were supposed to pass on our genes, or we were failed to do so, and then we haven't evolved to go any further. And so when we think about medicines generally, whether it's a drug for cancer or a drug for Alzheimer's disease or a drug for aging, we're really targeting this same problem, right? It's all one field, that evolution has failed to create us in a way to prevent these diseases because we haven't been selected in our 3.5 billion year history, or if you want to say Homo sapiens, our 100,000 year history, we haven't been selected to survive that long. And so the entire field of human medicine, in a sense, especially human medicine targeted at age-related diseases, is just accounting for this evolutionary neglect. And so I wanted to then kind of bring it back to Molly's overall question, which is like, how do we think about the work that's going on aging versus work that we already know and understand quite well, work that's going on studying cancer, studying Alzheimer's disease, et cetera. And to me, the fundamental difference between these two approaches deals with the stage that it's happening at. In kind of our traditional understanding of medicine, we wait for someone to get sick, and then it's only after they get sick that we say, okay, that person needs a medicine. But as our understanding of the molecular causes of aging that Bjorn was talking about has improved, and the tools that we use to measure damage in your DNA, or whether your metabolism is well regulated, as those tools have improved and improved, we've kind of moved the bar earlier and earlier in the ontogeny of a disease, right? Before it comes out and it's actually killing you, but is still causing some problems or about to cause some problems. And as we move that bar forward, a drug that a doctor working on treating your cancer stops thinking about, well, you have a tumor and now we have to give you a medicine so that you don't die, and it's moving in the direction of, well, you have these genome mutations, you're at high risk of cancer, so let's use the power of modern medicine and our understanding of biology to head that off at the pass, right? Do something about that before a person gets cancer. And that's, I mean, it's always hard for people to conceive sometimes what we mean when we talk about aging medicine and its intersection with pharmaceuticals, but I think that that's really it, right? How we use our knowledge of the biology of aging to change the healthcare system over a long period from one that waits for disease to happen to one that anticipates and prevents it.
So building on that point, just thinking about how we apply our knowledge of the biology of aging. I know that Bjorn and Jan, your research focuses on some of the genetic drivers of aging, including genome instability, DNA damage. So, given what James just said, which was a more of a focused area of application, more broadly speaking, what does this all mean about how much aging is under our control versus genetic?
First, the good news, okay? If you look at the animal world, then you see that certain animals, actually including humans, live very, very long. I mean, a bowhead whale actually lives much longer than we do. And some animals live very short. So apparently, evolution in all its wisdom over aeons managed to create the combinations of genes in a particular animal species to make it live very long. So in other words, to delay all those factors that cause aging. So obviously we can learn from that. That seems like fairly clear to me. Okay, that's the good news. The bad news is that it took so long for evolution to do. Why? Because it had to generate a very large number of genetic variation and play with that variation to come with new combinations of genes that gave rise to species that could live very long. So for practical matters, as James has pointed out, now we need to find those gene variants and try to manipulate them, make them druggable, as they say, sort of come up with drugs that can do the same thing as these genes do. Now, that's a big, big job, I would say.
What I find really fascinating is that when you think about, so aging is a property of our body, essentially, invariably, and not only humans, every other species has kind of the same issue. But then there's one exception, and it's really fascinating that in every single generation, our germline is completely set to 0 age. So with every fertilization, an egg is exactly 0 years old. And that has worked for us humans now in almost 7,000 generations. And so there is something inherently about a rejuvenation process that happens in every single generation. And I think we can learn a lot from that, how aging can be actually targeted, and how youthfulness in a cell can be reinstated, and maybe that can inform us about how we actually can augment the functional maintenance of tissues.
Based on our specific understanding of germline cells.
For example, I think this is a fascinating aspect, and we know now, for example, with all the technology of induced stem cells, induced pluripotent stem cells, that actually it is feasible, or it is actually at least not inconceivable, that you could actually regenerate tissues and organs.
Because what I'm going to say is immediately responsive to what you say. Look, I mean, yes, there's always been the question, if something in your cells, for example, call it DNA damage or protein damage, is causing aging, how come that our germ cells are free of aging? Otherwise, we wouldn't be here, not? I mean, germ cells, they're always okay, and they can give rise to new individuals every generation. That's what you just pointed out. Why is that? Actually, that's not true, not really true. In fact, there's an enormous amount of damage in your germ cells, and most germ cells never make it to fertilize an egg. Yeah? So what you see then, those lucky few who manage to get a fertilization, you got a beginning of an embryo, a conceptus, will go down the drain very quickly. We all know that. There are lots of very early abortions. And if you open up, if you study those abortions, it has been done, these early fetuses, then you see they're full of damage. So what you actually see here is selection. There's the power of selection. When you have germ cells, you can select out the best. When you have fertilized eggs, you select out the best. And even then, even those who are healthily born, they still carry a number of what we call knockout mutations in certain genes. So a number of genes don't work. Sometimes that shows up very late in life in the form of a disease. So in fact, there is damage there, but if you talk about germ cells, they have the power of selection. Most of our tissues don't have that.
Yes, of course, it's not going to be easy, but if you could integrate selection in the generation of pluripotent cells, that could probably answer to the concern of the damage and the selection.
I have a brief scientific vignette to talk about kind of, jumping off from where Bjorn is getting from. Thinking about the germline, I think, Molly, your question was quite apt, especially when we're talking, with some basic scientists on stage, right? How much can we learn from the study of nematode worms or germline stem cells or something like this? And I just kind of wanted to paint a little picture of how I've seen something that started from a very, very early basic science stage and is actually now at the point where it's starting to change some of the ways that we think about therapies. So a friend of mine studies fruit flies, right? Which you see kind of, when you have a rotting banana, the fruit flies come and like to land on it. And she studies specifically the testes of these fruit flies. So she spends all of her time cutting these flies into tiny little pieces to study their testes so that she can look at the germ cells. And the reason that she does all of this is because the fly testes is a quite simple relationship. It's got what they call a hub and a hub. A hub, so the hub is where the stem cells kind of attach to. And then just like spokes of a wheel, as new cells grow out of the hub, they make their way down a channel to become sperm. And she wanted to ask a very key question related to the study of these germline cells, because the germline cells do a very good job maintaining their genome. They can divide over and over and over again throughout the life of the fly. So what is it about these cells that keeps them so unique? And she made a very interesting discovery, which is that when a cell divides, it makes new copies of a lot of things, right? It makes new copies of its DNA. It makes a new copy of a lot of the proteins and organelles within the cell. And one of the things that it makes a new copy of is called the spindle. This is kind of like the anchor of the cytoskeleton. And she found out that even though the DNA, the new and the old DNA is kind of the same in the daughter cell and the old stem cell, the daughter cell always gets the new spindle. And this was kind of like one of these key things where now, as we think about some translational work that I worked on as my PhD student, we kind of jumped off of this and said, like, okay, well, if we can actually study what it is about that spindle that keeps the germ-like properties in this cell, then we can start thinking about ways to grow stem cells outside of the body in a regenerative tissue. And like, it's these little, and so I know it's a small vignette, but it's these little sorts of scientific insights that happen on the most basic level, sometimes studying model organisms, that can cascade forward, informing us of what questions we need to ask, what things we need to be looking at in order to imagine and develop new therapies. That, I don't know, it just felt like it was useful to talk about.
No, it definitely is, and it actually segues to my next question. So, longevity, you hit on longevity, spindles, right? What does contribute to longevity?
There are many mechanisms. So the caveat with the aging process is that it's complex. It's not only one molecule that would need to be young and the aging issue is solved. It's a complex it's a complex process, but we know what makes us old, and that's damage. And in fact, there are people that age much faster than we are. So there are these are genetic diseases, progeroid diseases. So where children are born with one genetic defect and they age within the first decade of their lives. So when they're teenagers, they look like 80 years old, and they die of, for example, of atherosclerosis. It's something no child ever gets because it's a typical disease of the elderly. And so the cause of that, the one single error that they are born with, are genes that repair their DNA, repair their genome. And so we know very well that if you don't repair the genome, you age in an incredibly high speed. So fast track to aging. And so we know that these repair mechanisms are so essential to give us the lifespan that we can enjoy. However, improving that further than the set of these genome maintenance genes that we have, improving that further is so far still very, very difficult, because it's really geared to the needs that we humans had in all our past. So that's a huge challenge, to actually improve repair capacities. So instead, we need to go for lower hanging fruits here. And these are really stress responses. These are these very mechanisms that are responsible for why calorie restriction extends lifespan. These kinds of pathways are really targetable, druggable, and these are kind of the lower-hanging fruits. They don't solve the underlying problem of the damage, but they keep the body healthy enough to avoid disease at old age. And that's, I think, is the key aim that we should nowadays focus on.
So I want to hear more also about some of the potentially druggable targets here, but also bring it back a level, because I read also articles written for the lay public about science, about aging research, and A lot of the times, what's really interesting about those is the comments section, where people say things about, my grandmother lived to 95 and she ate 2 pounds of chocolate every week and red wine and tons of olive oil, or on the flip side, my grandfather lived to be 102 and he smoked cigarettes his whole life. So can we translate any of this to lifestyle just yet?
Well, okay, before I'm going to say
I know we are, but is that, can we? Is it, go ahead.
First, I was actually very happy when James was just arguing how important invertebrate model organisms are, like fruit flies and worms. I mean, a lot of people don't understand that, and they basically feel that it's a waste of your money. It's taxpayers' money. Why do you spend it on fruit flies? You need to go to humans. That basically shows they have no understanding of how science actually works. Everything that we have, virtually everything in modern medicine, comes from this kind of organisms in the past. I just wanted to have that. That's certainly also true for aging. Now, regarding interventions, I think there are 2 types of interventions. Let's start with the most important one, and that's, I think, also what James was referring to. We really want to live healthy. We want to live into old age in a healthy way. That's the most important thing. We don't want to live until 60, become really totally degenerated and dysfunctional, and then live another 30 years. We don't want that. So that's the most important thing. The most important thing is really that we manage to develop interventions that keep us healthy for longer. Then old age is fun, I would say. Okay. So now so again, the good news is we actually are, I think, very close. You will probably say a little bit more about it. We are probably very close in getting there. There are a number of new drugs and other types of interventions that are now it looks to us that that's exactly what they're doing. We need a little bit more time to test them, but that's where we are. The other question you can ask, okay, but can we then live until we are 150 or so? That, I have my doubts about that. I think that's going to be very, very difficult. So in other words, more and more people will probably be able to live until they're over 100. We already see that. Centenarians is almost like an epidemic, if I may say. may use that. So you see lots and lots of centenarians now, and many of them, fortunately, are still healthy. That's where we want to go. But do we see people over 120 or so? No, that's extremely rare. In fact, as far as we know, there was only one woman who ever managed to become 122. And I was actually at an economics conference in the south of France, nothing to do with it. And then there was a guy next to me who introduced me, and he was asking what I was doing, and I was talking about aging. He said, "Oh, you know about Jean Calment?" I said, "Well, yes, she's the record oldest living individual." He said, "No, that's a fake. She was actually her daughter." Okay. He said, "Yeah, because the guy who studied this work in France, he was a college roommate of mine, so we talked about it in the bar." Again, I don't say that that's actually true, because I don't know. That's what I heard, but it tells you a little bit about things are not always as clear as they sound. All right.
I'm glad that you said that. It totally takes the wind out of the sails of what I was going to say, because to Molly's point, the story of this woman, Jean Calment, who's the oldest living person on record, allegedly. So the story about her is that she quit smoking when she was 99. And then she quit drinking when she was 117.
And that killed her.
And that killed her, yeah. But to your point directly, when we think about all of these examples that we're seeing, centenarians, which are certainly becoming more and more common, but also these anecdotal stories of, like, you know, my great-grandmother lived to 105 or whatever. Not my great-grandmother, unfortunately. I have a lot of disease in my family. All died early. But, you know, so-and-so relative lives to a ripe old age and was healthy and so on. There are 2 aspects at play here. The first is that there clearly is a genetic and also kind of nurture-based component to longevity, right? So I guess 2 quick stories. If you look at groups of like families, so there are long-lived pods of families, like it wasn't Jean Calment, but another group of centenarians where one member lived to be a super centenarian, which is above 110 years old, right? And all of her 6 brothers and sisters, they also all made it past 100. And so you see evidence like this cropping up over and over again, where there's clear genetic components. And when researchers like Jan, goes through a huge amount of data, certain genes and gene polymorphisms start to appear as being helpful in making someone more likely to live to 100.
Could we also add the piece of information that, as to date, we know that 20% of the variance in longevity is explained by genetic variants? I just think it's important to tell the audience, yes, this is all very important, but so far it's explaining 20%.
And that's exactly my point. It's a small percent of the variance.
But that's only, say, when you are capable of living to a very old age, then the genetic component is much more important.
Well, of course, but it's a highly selective group.
Well, sure, but that still will...
It's interesting to study, that's fine.
Right, because that will still tell you how these people manage to get so old.
This group.
Yeah, that's why. Yeah, this small group. Sure, but that's why. But if you look at mortality in general, that includes like car accidents, for example. So if you look at genetic component, yeah, then it's only 20%. But if you now look at those people who really manage to become older than, let's say, 60 or 70 or 80 or so, you see that the genetics becomes more and more important. So it's not everything, of course. Well, of course, it's not everything. But it is. I agree with James. It's a major component of extreme longevity.
And can we learn something from that 20% and apply it to the other 80%?
That's precisely what we think and what we are trying to do now. Yes, as James said. Yeah.
It's a big challenge though, because you have, even though it contributes in a major way to this very old age, it's a smaller effect as you kind of go down and you look at what differentiates someone who lives to 70 to someone who lives to 90. The genetic components are much less clear. But as we kind of take our lens back and look at what the mice and the worms and the flies are telling us, there is a story here that is much more consistent. And this story is one where if we can do something about the various types of damage, and instead of taking a backward-looking approach, looking at the longest-lived humans and coming back, which I think is a completely valid and interesting approach, if we instead start with our types of damage that we understand, the things that go wrong with aging, and then almost approach it like an engineer and build up from this, saying, if we fix this one thing, what happens? And if we fix this other thing, what happens? How about if we combine those 2 things that we fixed together? This is, I think, part of what the magic of this space that has kind of occurred over the last 3 to 4 years, because as a field, we've made a really fascinating discovery, which is, I think, when I got into this field 15 years ago and was talking with some folks, we all expected that since we knew that there were, depending on who you ask, anywhere between 5 and 15 different types of damage, things that go wrong as we age, most people would have said if you fixed just one of these things, people are still going to get old, they're still going to die, and it's not going to make that big of a difference just affecting one of the, let's say 10, one of the 10 things. But the reality is quite different, because as Bjorn talked about, you can change just one growth factor or eliminate a few bad cells in the body, and all of a sudden it changes the physiology of a mouse so much that it can live 20%, 30% longer. And that's always sometimes hard when I talk with people to kind of grok what that means. Like, what does 30% longer mean?
Right. What is that?
Right?
I was going to ask you that.
Yeah.
Because do you mean 20 to 30% longer than your life expectancy, or 20 to 30% longer than what we think is the maximum lifespan of a particular species.
That on average, that group of mice tends to live.
And that's exactly it, right? So the equivalent here would be if we right now have an average lifespan as a society of about 80, right? 10% of that lifespan would be 8 years. And so if we lived 30% longer, then that could increase the average lifespan from, let's say, 80 to, let's say, 104. Is this realistic? Well, it works in mice. We'll see whether it can work in humans.
Don't really know if that works.
Oh?
Not really. I mean, the only thing you can clearly see is that the average lifespan of a mouse, for example, we talk about dietary restriction, as Bjorn mentioned, which is, I think, still the most convincing way of increasing lifespan. You can see that the average lifespan increases, but let's say you have 50 mice, and you subject them to caloric restriction, and then you have 50 control mice, yeah? And then you see at the end that the oldest mouse in the control group is only, let's say, 30 months, and in the dietary restriction group is like 36 or 37 months. So then you would say that the maximum lifespan has changed. I would not say that. I would say simply because the mouse in the dietary restriction group were healthier, they had a higher chance to survive a little longer. But I have no clue if I actually changed the actual maximum lifespan of the species, because we don't even know what it is. How do you know what the genetically determined maximum lifespan of a mouse is? You need to study 1 million mice or so, and then wait until the oldest one dies, and then you would know. Like we know for humans. For humans we know it, but that's the only species.
I'd like to come back to this, the point that was raised from the audience, the question is aging a genetically controlled process? Does that mean that with the genes that we are born with, do we have a set expiration date? And that is clearly not the case. So it's more like we have a rough shelf life that is now about 80, but it is under great influence on environmental factors. And that's what we learned, for example, from twin studies where two identical people, if it was solely determined, so if the genes set kind of a clock of aging, you would expect that they all fall dead at the same time, but they're actually not. And by 2/3, they have different lifespans. So that means that there are environmental influences that are quite substantial. And I think this is the first good news, that we can actually, by modulating our environment, we can actually change a lot of that. So by dietary interventions, by lifestyle, like sports and all these things, there are many things that can change. And what James has also alluded to is that the centenarians, they teach us one really important lesson, and that is that a significant portion of those people that really live to a very old age, so they are above 100 years old, a fraction of about 20 to 25% of those don't only live that long, but they stay healthy that long. So they have their disease time, their disease span is compressed to 1 or 2 final years of their lives, which happens very, very late, so way above 100 often. And so they teach us this lesson that it is possible to stay healthy for a very extended period of time. And we have to figure out how we can make that possible for a large fraction of our population, because the alternative is that a large number of us will be really sick.
So what do we know from humans? Because from other model systems, we can look at lifespan maximum, or whatever you define it as. You can look at health span, but humans have the happiness factor too. So what do we know about that?
Well, there is a study, if I can just make this point. There are studies that suggest that people who have a positive view on aging live longer and stay healthy longer. Now, that's a correlation. There's not no causation behind that, because what if just those people who have a positive impression of old age happen to be in a family where people stay healthy for a very long time? So the happiness factor can be a consequence as much as a cause. We don't know that.
Yeah.
It's a little bit complicated because I think it's important to make clear what the difference is between life expectancy at birth, that's simply the average lifespan, including like when you are unfortunate and you get a car accident or so. Those people are included in that set. The other thing is the maximum lifespan of a species. So how long can a human maximally live? That means that you have to look at the oldest one, the oldest recorded humans, and then see how that is. You can do that for a mouse, but the problem is we don't have enough, because you pick 100 mice, look, it's the same as humans. You pick 100 humans, and in that group, the oldest one is, let's say, 75 or so. You're not going to say then that the maximum lifespan of the human species is 75, because we know it's over 100. But in that small group, it was the longest lived individual. Could have been an older one or a younger one, we don't know. So that's a very important thing, because that tells you what our genetic mechanism is as a species. How long can we really live maximally? I think we need to understand that, because there's no doubt that we can improve the average lifespan. We can do that in animal species, and we can do it in humans. This is the most important thing. The human life expectancy at birth, only like 200 years ago, was maybe 25 or 30. Now it's 80. So we made enormous progress there, and I think, and I hope, that we can make a lot more progress. That is what we want to do. That's the health span. But it's still scientifically very interesting, and maybe for some people who want to live forever, it's also interesting. Can we break through that barrier, that genetic barrier of our species? Can we live until 150 or 200 or 250? That's a whole different question, I think.
Good evening, everyone. I used to work with Jan in his lab as a postdoc, and I'm the wife of Björn Schumacher, with whom I do not agree with you tonight. Because Jan and me, we do agree on the effect of epigenetic, and here where Björn, I do not agree with you. Happiness is a causality, because if you formulate it as a happiness, of course, you don't have a scientific biomarker, but if you formulate it as a lack of stress, therefore, here you can see it as, these people are less stressed, they are more relaxed, therefore they have less comorbidities. That's all what I want to say.
The other side is that people who have been massively traumatized tend to die prematurely, about 5 years compared to the rest of the population. So that's the other side of the same.
Stress factor, yes.
Let's wrap up let's have a little more panel before we open it up.
So I want to hear a little bit, kind of focus in a little bit on more disease specific. So let's hear a little bit about cancer, which is an aging-related disease. So what does a cancerous cell do so right that it can survive against all odds? Are we, given everything we just said about what we can control versus what we can't, are we giving cancer cells a good home? Is it a whole bunch of random stuff in the wrong direction stacked up over time, or are there particular traits of cancerous cells that make them successful?
So yeah, cancer cells are enormously successful in kind of taking over our body by becoming essentially immortal. I mean, cancer cells, you can grow infinitely. So they kind of have a mechanism of immortality, but then they are very lousy at doing any functions that we actually require. Like, they are not functional nerve cells or functional intestinal cells, but they are just good in growing and growing and growing forever. And but it teaches us really a lot about the mechanisms that usually restrict immortality of cells. Because it's complete, there are checkpoints on every cell that prevent them from becoming cancer cells. And these mechanisms are kind of inactivated, switched off by cancer cells, and so they just by default become immortal. Now, what's really fascinating is that when we talk about interventions of aging, what is really extraordinary is that when you look, for example, at dietary restriction, which we've known for a very long time to be really a universal lifespan extension method, what it is really good at is reducing cancer risk. So it's very possible that when we think about preventive therapies, so if we think about we want to extend health span, target the aging process, and one disease that I believe we can be most successful with is really reducing massively the cancer risk by interventions. I think this should not be underestimated. And I don't think we will solve cancer just by treating cancer, but actually also by reducing the risk. And I think from the aging field, a major innovation there will come.
Jumping in here, so the way that I think about cancer is derived a lot from the study of one of my favorite fields in biology, which is host-pathogen interactions, right? How a virus or a bacteria interacts with someone that it's infecting. And if you think about what's happening with a bacteria or a virus, it doesn't care about the overall health of the organism. In a long-term way, it does, and we can get into the details of when it's true. But if you look at something like the Ebola virus, right, that just comes in and starts replicating, and its only job, evolution is just pushing it to say, outcompete, produce more, copy yourself again and again. And that law of evolution is present at all tiers of biology. So, different species will compete with each other, different groups within a species will compete with each other, and within an individual of that species, all of their trillion cells are competing with each other because they're driven by that same law. So no matter what our species-wide evolution has done to control those cancer cells, each individual cell of our body is being pushed, nudged subtly by evolution to divide, to outcompete its neighbors, to become cancer. Cancer is evolution succeeding within our body. And by the time, as Bjorn was talking about, there are so many checkpoints that we've got built in to prevent our own cells from becoming their own immortal evolutionary creatures within us, that by the time we actually recognize cancer as a disease, it's jumped through 7, 8, 9 of these hoops, right? It's overcome barrier after barrier after barrier, so that by the time it's ready to threaten us, it's incredibly evolved and very good at dodging things that we can do to it. And I think that, so Richard Nixon started the war on cancer in 1974, right? Way before we understood this much about cancer biology. And I think that one of the fundamental, I was gonna say failings, but let's say just major challenges about thinking about how we study cancer and how we've approached it so far, is that no matter how innovative our cancer therapy is, it's just one more thing of the 7 things that tumor has already encountered and overcome. And you do 2 interventions, and now it's 8 and 9 things. And the approach from aging biology is remarkably different, right? Because it's changing the way that the cancer is evolving. So touching onto that tumor at stage 1 or 2 of its evolution instead of at stage 8 or 9, when it's much more vulnerable, when you can, just by slightly changing the way that your body is surveilling its own systems, you can kind of clamp down on those early stage tumor cells. And this is why I think we see this incredibly powerful effect of treatments targeting the aging process on tumor genesis.
So I'm just mindful of time. I have one more question, and then we're gonna, I think, turn it to the audience because I have a feeling we'll get some good questions from you guys too. So James, this is for you, just because I'm really interested to hear more about your work now, because you aren't currently doing your own research, but you're actually enabling people who are doing research to get therapies out faster. So, given everything that we've just talked about and, the huge field of aging research, how do you know what looks good? Like, how do you think about that? And is there like a secret sauce that you look for, or how do you strategize based on that? Because there's so much research out there and it's moving so quickly.
Sure, yeah, there really is a lot happening very, very fast in this space. And there are really great labs like Bjorn and Jan's lab around the world in an awesome network of aging scientists that are all, I think, making discoveries at a pace, especially in this field, that we've never seen before. The way that my group looks at things, it is, we see ourselves as one piece in a long chain of trying to deliver something useful to a patient. And so, as I kind of alluded to before, that starts in a lab where a scientist has an idea about some interesting part of biology, and then it might go for 10 years being explored in that biological laboratory, and eventually it reaches a stage, and not all research is ready at this stage at all times, right? But something will kind of pop its head up and say like, hey, this is something that might be applied to a human. And so we're kind of surveilling over this field of aging biology, looking for those little buds popping up so that we can nurture them. And I can tell you like a couple of, I think maybe it would be useful to just have a story about one of the projects that we did go with that kind of emphasizes this. So one of the strongest, we talked a lot about genetics, but there are actually also pharmaceutical compounds that, like pills that you can take that extend the lifespan of a mouse. And one of the most potent one of these is called rapamycin. And rapamycin, we've known about it for some time. I think since the early 2000s, the first mouse studies emerged, and it works in nematodes and in fruit flies as well. And with rapamycin, you can give a pill to a mouse and it lives longer, healthier, less incidence of cancer, better memory, it'll run more on a treadmill, all sorts of things. It's a lot longer too, like almost 20% works for both sexes. But there's a problem, because rapamycin it's an FDA-approved drug that has a different use. And its main use in the clinic is to suppress your immune system when you're getting a kidney transplant. And so this is not a drug that any of us would want to take on a day-to-day basis. If you're a mouse in a sterile cage in a laboratory, then okay, maybe that's fine. But you don't want to take it on a day-to-day basis. And so one of the things that we looked for, because we knew about the power of rapamycin, it was clearly one of these buds that was sticking up as like, hey, mTOR inhibition, which is the pathway that this goes after, this is something very important in aging. And so we looked around for a group that was really thinking about how to work on this pathway in a way that could minimize the side effects that we see on the immune system, and there's also side effects on metabolism, and make a safe version of the drug. And so we ended up partnering with a professor from the University of Wisconsin who had discovered that this rapamycin molecule targeted two pathways. One that causes the mice to age slower, and a second one that causes depression in the immune system and metabolic side effects. And after he made that discovery in 2012, after a career of work, right? After he made that discovery in 2012, that was the light bulb that activated us, right? Where it got to the point where we could, and of course, my firm wasn't around back then, but they started a company. We then supported that company since the beginning of Apollo. And they were able to create the first version of this rapamycin molecule that doesn't have those side effects in mice by just targeting the first pathway and not the second, so that then we can use that molecule to treat people who already want to be taking rapamycin, not people in this room necessarily, because maybe eventually, but it's not happening right now. But there are people with genetic diseases where this mTOR pathway is overactivated. They need a molecule like this right now because they're suffering from the side effects of taking this molecule today. But then in the long term, after we can show that our new molecule is safe and effective, then we can maybe, hopefully someday, use it to prevent cancer, prevent Alzheimer's disease, slow the aging process in humans.
All right, so 15 seconds each, 15-ish. I don't actually have a timer, so I'm gonna eyeball it. And then we're gonna go to the audience. What's the #1 breakthrough in aging research and or application that you'd most like to see?
To understand the causes of aging. We don't know, we really do not know that. It's an interesting thing to say, well, there's all sorts of damage, sure, but what's primary? What's really, is there a cause? Is there really one basic cause that causes all the other things? Is that known? We don't even know that. That, I think, is the major thing that we need to figure out.
I would go for we really should try to understand how naturally rejuvenation works, which works wonder in every generation of us. And I think we don't understand how that happens, how that is possible. We understand some aspects of it. But I think there we will learn a lot about aging and about how to keep cells functioning and in a good shape, and eventually the body in good health.
Mine is a little bit more practical and on the clinical side, which is that groups like mine and research groups like your guys, we can make drugs that have the potential to extend healthy lifespan. And it looks from the evidence that we have so far that we can actually make a lot of these molecules. But before these things can be shown to be safe and effective in humans, that's going to take clinical trials. And doing a clinical trial for preventing the diseases of aging, if we don't have any extra tools to do that, it's going to take a very long time. We're going to have to give someone a pill and then wait 20 years to see if they get cancer. And so I think one of the biggest watersheds in this field is going to be the development of the tools to detect whether somebody has changed their risk profile for the diseases of aging in a way where we can do shorter clinical trials to test whether these anti-aging drugs are actually working or not. And I think that that's going to be kind of the key watershed before these enter broad use.
All right, so audience, what have we not talked about?
Thank you so much. It was extremely interesting. My question is a little bit off the wall, and I hope you'll bear with me. Are there any lessons, all of you have studied aging and what makes for a long life or doesn't. Are there any lessons, philosophical lessons, that can be applied not to the human body, but to forms of government, society, the longevity of the government in Rome? Are there any parallels, or in the United States, or in Germany? Are there any parallels that can be made philosophically from the human body to society? And forgive me if it's a little off the wall.
Well, I will try to say a little bit about that. I don't know if I really answer your question, but of course, since there are now so many older people in good health, I think it's very important to understand better how that generation really differs from earlier generations in their behavior, in their desires, in also their capacity to think and to work. I mean, it's clearly a myth that, for example, when you're older, you are less creative. There's lots of evidence that that's just not the case. So, but of course there are differences because you grew up in a totally different generation. That is something we've never before in history experienced, that we have this kind of big differences between generations. For the luxurious problem that you have is we have all these healthy, very old people now. So that's very important, I believe, for government programs to be developed to understand that and to appreciate that. That's one thing. A very practical thing, of course, is that we need to dress up cities and buildings in a smart way so that older people who necessarily have a degenerated musculoskeletal system, I have that, for example, it's very useful to have all sorts of safeguards, not so many curbs and all sorts of ways so that they can walk around easier. That's something that government should do.
I have to kind of to address your philosophical point, as what I suspect you're also a student of history. There's an adage that if an empire is not growing, it's dying. And I think that if we wanted to extend something on the more philosophical level, the human body is similar, right? We grow and we go from one little point of light, one little cell, one little town, into an empire, and then, or a body, and then at that stage, once it's fully grown, maintaining it is a hell of a challenge. You can't grow much bigger. There's some kind of wall there. But maintaining something that has grown out and is so complex with so many interconnected parts is an enormous challenge. And so if I were to make a philosophical equivalent there, I would say that creating an immortal cell, right? A single town that exists since Roman times, that's easy. Creating a whole organism is going to be much more challenging. There are ways to do it, being thoughtful. I mean, we can talk about extending the philosophy, but I just kind of wanted to create that analogy between your governments and empires to a human and longevity.
Thank you.
I have an auntie who is 75 years old, have no gray hair whatsoever. And also I saw people who have gray hair, but no wrinkle at a very old age. So can we learn from those people and research maybe their gene have something we don't have, maybe we supplement ourselves. Thank you.
Yes, absolutely. I mean, we can learn a lot from people who maintain their good health in very old age. Now, wrinkles are not the best example because wrinkles are particularly problematic for Europeans because of the way our skin is made. We have a very thin dermis, so we're a bit thin-skinned, and that's why we wrinkle much earlier. So
they're also quicker to be offended about wrinkles.
But it's quite amazing how much attention people pay to the outermost marks of aging, and I think we are way too, like if you tell people you work on aging, they immediately ask you what's the cream that I can use, right? And but that doesn't change your inner state of age. That doesn't make your liver any younger. So cosmetics is the wrong thing for that. Might be other things to keep livers in better shape. But I think we can learn a lot from those people that become very old, particularly centenarians, that stay in very good health. That tells us it can work. We also have there are studies that actually show. So on old people, there are actually very little medical studies done. But there's one interesting study recently in the Netherlands where they found actually that interventions can even have positive effects for people above 70 years of age, and very conventional types of intervention at this point, because what James has described with the new drugs, that unfortunately is not up and running as treatment options at the moment with sufficient evidence. But these dietary restrictions combined with a workout, a healthy lifestyle with regular workout, can actually have positive effects even when started at old age. So there's a lot of things that can be done to stay healthy. But that being said, it's still not enough, because we really need these interventions. We need preventive medicine that require a whole paradigm change in medicine, because we will be needing to treat healthy people to maintain their health. This is something completely different than what medicine does nowadays, waiting for the disease to come. And if you're above 60, you're more than half of those people above 60 suffer from multimorbidity. That means they have at least 2 chronic diseases at the very same time. So we really need a change. We need a policy change, because we need to be able to target the aging process. We need to change the way clinical trials are being conducted on that. And so it really goes way beyond the science that I think is on a very good way. But we also need policy to really develop that.
I think still it's an interesting question, because what you basically say is that there's individual variation. Not everybody ages the same way, and that's, I think, one of the best characteristics of aging. There's enormous variation. I mean, you mentioned hair color. I mean, I lost all my hair, but Ronald Reagan also, I believe, never got gray hair, not?
And tissue variation.
And
tissue variation.
Yeah, there's variation in the rate of aging on a tissue-by-tissue basis. Yeah, that's also the case. But of course, what you can see outside, how your skin looks, that's visible for everybody. So that's why people pay so much money for it.
Let's take a couple more questions. I know I saw some hands in the back.
They're constantly, they brag to me that people in Japan and Hong Kong, which have been prosperous for a while, live longer. They have the longest lifespans. Is it possible that there's a genetic basis for this, and that there's differences among populations, and that you don't have to just study fruit flies?
So I can jump in, because to repeat the question for those of you that didn't hear it, the question is, people in East Asia and Hong Kong and Japan have the longest average lifespans right now of anywhere in the world. So are there genetic components to these entire population regions? And there's a great study on this, because there are areas called blue zones around the world where there are way, way, way above average numbers of centenarians, right? A lot of more people living above 100. And one of those areas was in Okinawa, Japan. This is one of the strongest ones. And the people in Okinawa practiced a tradition called Haru Hachido, which is the rule of 80, something like this, or the rule of 8, where whenever you have a plate of food, you eat 80% of that food, and then you leave 20% as tribute to your ancestors, which is very much like the caloric restriction model that we know here, and that Bjorn was talking about. And so it turns out that if you take these long-lived Okinawan populations, there was an exodus of a large group of this population to Brazil. And one generation later, looking at the descendants from both genetically nearly identical groups, Okinawans in Brazil, who had adopted a Brazilian diet, and Okinawans in Japan, all of a sudden, the Okinawans in Brazil had no increase in centenarian number. And it was entirely driven by the environment and the diet. So disentangling these things in human populations is exactly why I place so much weight in the use of animal models to do this, because if you're relying on natural experiments over the course of generations, then the science is really limited by the best natural experiment that you can find, which, just like if you study economics or social sciences, is always a complex web of interfering factors. But I mean, I'm a mouse geneticist by training, so I guess I have a bit of bias.
Calling on me, and it's a wonderful discussion. And along the dietary lines, my question's related. There's a doctor in Atlanta, Dr. Lila Denmark, who lived to 111, and she actively was a pediatrician treating her patients until she was something like 103. And she had very strong theories about cows, that cow's milk is for cows, that once you are past your infancy, that's it, no more milk. Any kind of product coming from a cow interferes, as you said, with your life expectancy. And it worked for her. So could you weigh in on that, please?
Well, I'm from Holland originally, so I should be a milk expert. I actually, honestly, before I came here, I actually drank a glass of milk. I like milk. But I don't think this is related to a long life. In fact, humans are really advanced machines, and nutrition doesn't really have such a major impact. People think so because, well, there's so many of these nutritional supplements, but in reality, it really makes not so much difference. Yes, caloric restriction, that certainly should do it. And of course, when you eat only French fries and hamburgers all your life and nothing else, I'm sure you will die from a heart attack very early. But in general, people shouldn't exaggerate the impact of diet. I don't think there's any major evidence for that. It's the same story as what you just heard from the blue zones. There's always, as James pointed out, there's usually an easy explanation for that. It's not so that there's like a group of holy people who have particular genes. It can't be. I mean, our species, humans, are too close. They are very closely related to each other. There's absolutely zero evidence that a certain group suddenly developed like different genes. They don't. So all of us are all sort of the same. It's not the case.
A couple more, right in the front here.
Very much enjoyed the discussion. A question here, we talked a little bit about the use of different animal models, so mouse models and Drosophila flies. What can we learn from the use or the study of species that are known to live a very long time? I think about turtles, or maybe there's some other more complex animals that have far longer lifespans than we do.
That is indeed very interesting. So it's particularly valuable to study species that are very closely related and achieve naturally very different lifespans. And one of those species that has recently become focus of quite active aging research was the naked mole rat.
The cutest animal that you'll ever see.
It looks as the name says. The name exactly describes how it looks. But that doesn't prevent it from living way longer than related species. And so it's indeed very interesting to study these species. There's also, for example, a very interesting new model system, which is a killifish that there are many different species of killifish in African lakes, and due to their ecology, these different species have vastly different lifespans. And so people started now, only recently, to study these animals. I think biology always needs to simplify somehow, because biology is so complex that we always trying to have simple animal models. And we only have really a handful that we studied thus far. And I think with the modern technologies of gene editing, of genome sequencing and things like that, we are now in a position to greatly expand that. I mean, of all the millions of different species that exist, biologists really study in deep detail only a handful. And that is being expanded currently, and I think there's a lot of new knowledge and insights that we can gain from that. And never forget, things rarely get invented twice. So what is true, as Jacques Monod said, what is true in a bacteria is also true in an elephant. And that is true throughout biology. So it's very relevant to study different organisms. We need to expand model systems, and this will have major impact on our understanding of the human body.
Can I make one quick comment on that? Because it's sort of interesting. I was actually today working on a grant application of one of my students who is actually working on that species difference between, as Bjorn just said, naked mole rat and mouse. A naked mole rat, they're about the same size and very closely related. They're both rodents. But the mouse lives about 3 years in the lab, and a naked mole rat at least 30 years, probably more. So 3 months, sorry, the mouse 3 months, naked mole rat 30 years, probably even more, we don't know exactly. So she takes cells from these two species, treats them with the same dose of a DNA damaging agents, and then sees if the naked mole rat can repair the damage much better than the mouse cells. And her results clearly show that that's the case. So here you basically see this in action. We basically work with species with many big differences in lifespan, and we do indeed see that the longest lived species, their cells are better able to repair.
Thank you very much for a very exciting discussion and giving us some insights into the complexities of the biology of aging. I'd like to clarify that we heard a lot about the biology of aging, even though for shortage of terminology, we often say aging. It's the biology of aging. And one of the most fascinating things for me, you were asking that question in your first round, what's the most fascinating thing for me as an aging researcher is that we as a species were prepared by evolution to change ourselves, and we have done so. We have expanded our average lifespans tremendously over the last 100, 200 years, and most likely not by doing the work of evolution, but by epigenetic processes and by influencing our environments, engineering our environments, and by influencing our lifestyles, and by influencing our values and attitudes. And we are just at the cusp of getting a better understanding of these complexities. And my pledge is, as we are getting more and more tools in the biology of aging, in the psychology of aging, in the social sciences, and in engineering and technology, that we find ways to put all the modules back together, rather than, because each of it is so complex, and you were saying, okay, we're only touching the surface, and we spoke about genetic repair. We didn't speak about epigenetics, microRNAs, telomeres, and all these other things. That's only biology. And then I can start about psychology. We heard a little bit about effects of stress, and we heard a little bit about the effects of physical activity, and I could go on and speak about the effects of work environments that we expose ourselves to and how we can change these. But there is no place right now in the field of aging research that undertakes the effort to put these different atoms of aging insight back together. And of course, it's a major effort because as we specialize, our languages diverge and it gets harder and harder to speak to each other. But it seems to me that the future of aging research is by putting the things back together. And I mean that in an understanding that does not consider aging as a disease.
So much. You know, we are pretty much out of time tonight, but I want to invite everyone to continue the discussion and bring your other questions. Is it downstairs? Do we have a reception? Thank you so much for a fantastic conversation.
Yeah, thanks again to our wonderful panelists and our wonderful moderator, Molly, and the responses from the audience.